Usually when we talk about a medical diagnosis,
Yeah.
there's this comforting expectation of precision. It feels almost like engineering,
Right, yeah. Like a mechanical issue.
Exactly. You break your arm, the X-ray shows that jagged white line,
Right.
and the doctor just points to it and says, there it is. That's the problem.
It's binary, it's visible, and therefore it feels solvable.
Totally solvable. Broken or not broken. It's super clean.
Yeah.
But then you step into the world of neurodegeneration, and suddenly that X-ray machine is just useless. We're looking at a diagnostic landscape that is incredibly murky.
Oh, absolutely.
You don't get a clean break. You get a slow, devastating fade.
It really is the absolute definition of diagnostic muddy waters. And historically speaking, very few diseases exemplify those muddy waters, quite like amyotrophic lateral sclerosis, or ALS.
Right. And that is exactly our mission for this deep dive.
Yeah.
We're unpacking a genuinely dense, but incredibly groundbreaking paper recently published in Nature Neuroscience. This is from October 2025.
A really massive paper.
Yeah, massive. And the goal here for you listening is to fundamentally rethink how we understand ALS. Because for decades, the medical community has basically been staring at one part of the map. And today, we're shifting the spotlight entirely.
We really are. Traditionally, when we think of ALS, which is this lethal adult-onset motor neuron disease that just slowly robs a person of their ability to move, speak, and eventually breathe, the focus has heavily been on the central nervous system.
Like the brain and the spinal cord.
Exactly. The assumption has always been, well, the motor neurons are dying at their source.
But this paper looks at the absolute opposite end of the system. We're zooming all the way out to the peripheral nervous system. Specifically, we're looking at the neuromuscular junction, the NMJ, and the muscles themselves.
Yes. The NMJ is the exact point where the very tip of the nerve ending meets the muscle fiber.
It's the literal connection point.
Right. It is this critical microscopic synapse that allows your brain's electrical signals to actually tell your muscles to contract. And in ALS, long before the whole nerve dies, we see severe disruption right here at the junction. The connection just dissolves.
Okay, let's use a visual memory trigger here to get this planted in our minds. I want you to imagine a massive, complex power grid.
Okay.
For decades, when looking at ALS, scientists have been intensely studying the central power plant, the spinal cord, trying to figure out why the lights in the city are going out. But ALS isn't just a failure at the central power plant.
No, it's not.
What this research shows is that there's a catastrophic breakdown happening at the specific outlet where the wire plugs into your house. That outlet is the neuromuscular junction.
That's a perfect analogy. The power plant might be struggling, sure, but the wall outlet is actively melting down.
Yeah.
And to understand why that outlet is melting down, we have to introduce the prime suspect of this whole investigation.
Okay, lay it on us.
It's a protein called TDP-43. Now, under normal circumstances, you have to understand that TDP-43 is a vital, hard-working molecule.
It's not inherently bad.
Not at all. It's a DNA and RNA binding protein that usually lives safely inside the nucleus of the cell. Its day job is basically acting as a librarian, helping to regulate genes, process RNA, and package it for transport.
So it's a model citizen of the cell. It's supposed to be there.
Exactly. We need it to survive. But in ALS, TDP-43 goes rogue. It mislocalizes. It basically escapes the nucleus, floods out into the main body of the cell, the cytoplasm, and it begins to clump together.
And these clumps are bad news.
Very bad. It forms these toxic, sticky aggregates. In the scientific literature, they call them condensates. And it doesn't do this alone, actually.
Wait, that is an accomplice.
It does. It grabs onto a stress response protein called G3BP1, and together they form these massive, suffocating roadblocks inside the cell.
Wow. Okay, and here's the big question, the core logistical mystery I want you, the listener, to ponder right now. Motor neurons are some of the longest single cells in the human body, right?
Yes, absolutely massive cells.
The cell body, the nucleus where this librarian protein is supposed to be doing its job, is located all the way up in your spinal cord. But the axon, the microscopic wire extending from that cell body, stretches all the way down your leg to your big toe.
Right.
So if the neuron center of operations is in the spine, how on earth is this massive pileup of toxic TDP-43 protein occurring at the very tip of the nerve, thousands of physical miles away, on a cellular scale.
It's a complete logistical nightmare. I mean, if the cell body was building these toxic clumps and shipping them down the axon to the toe, the sheer energy and time required would be staggering.
It just doesn't make sense.
No. And that brings us to the first major revelation of this paper. To understand why this toxic protein is accumulating at the nerve terminal, we have to stop thinking about shipping and start thinking about local manufacturing.
Local manufacturing. Okay, let's ground this in the human evidence first, though, because this isn't just scientists playing with theory in a petri dish. They actually went looking for this in real patients.
Right. The researchers started with human clinical data to ensure this mechanism actually happens in real life. They took biopsies from the peripheral nerves.
Which nerves exactly?
Specifically, they looked at the obturator nerves in the leg and various intramuscular nerves. And they compared a few distinct groups. They looked at non-ALS patients who suffered from a different kind of nerve damage, just as a control.
Makes sense.
Then they looked at patients with specific inherited ALS mutations, like the SOD1-C.442G mutation, and finally, patients with sporadic ALS, which is the most common form of the disease that happens without a clear family history.
Okay, so they're looking directly at the biological crime scene, right at the far end of the nerve. And what did they see? Did they catch the rogue protein in the act?
They absolutely did. They found phosphorylated TDP-43, which is the toxic, clumpy version of the protein, forming these dense aggregates directly inside those peripheral motor axons in the ALS patients.
Wow.
Yeah, it wasn't just floating in the spinal cord. It was piled up right there at the endpoint, choking the neuromuscular junction.
Okay, let's unpack this mechanism, because this is where my mind got completely twisted around. If it takes too long to ship a toxic clump from the spine to the toe, then the nerve isn't shipping the broken cargo down a long-haul highway. The nerve must be building it on site.
That is the crucial paradigm shift here. The nerve isn't shipping the finished toxic protein, it's shipping the blueprint.
The mRNA.
Exactly. The messenger RNA, the genetic instruction manual for building TDP-43, is naturally abundant all the way down the axon. The problem in ALS is what we call aberrant local synthesis.
Meaning the nerve terminal has its own local factory. It's essentially 3D printing this protein right there on site at the NMJ just to handle daily maintenance.
Right.
But in ALS, that local assembly line gets stuck in overdrive. It starts printing way too much TDP-43.
Precisely. And the consequence of this localized overproduction is just devastating for the nerve. Remember how we said TDP-43 naturally binds to RNA?
Yeah, the librarian.
Well, when you have massive toxic clumps of it forming at the nerve terminal, those clumps act like a sponge. They begin to sequester essential RNA. They trap all the instructions meant for other critical jobs.
So it creates a massive molecular roadblock. Everything gets jammed up.
Exactly. Because all the RNA is trapped in these clumps, it completely inhibits general local protein synthesis. The nerve terminal suddenly can no longer build the everyday proteins it needs to simply survive.
Oh, wow.
And without those maintenance proteins, the local mitochondria, the batteries of the nerve, they begin to malfunction. Energy production plummets, and without energy, the neuromuscular junction simply withers and disconnects from the muscle.
Okay, let's take a beat and trace the logic here so we don't lose the forest for the trees.
Yeah.
Here's our first quick recap bullet.
Right.
1, the disease causes a breakdown at the muscle outlet. 2, the culprit is a rogue protein that clumps up.
Right.
And 3, the plot twist is that the toxic protein isn't leaking from the brain. It's being aggressively overmanufactured right at the tip of the nerve, choking the nerve to death.
It's the exact sequence.
Which instantly begs the question, and I want you to think about this. If you have a local factory that suddenly goes completely rogue and starts churning out way too much of a dangerous product, where is the factory manager? What is supposed to be regulating this local 3D printer under normal conditions?
And this is where the paper stops being just a study of ALS and becomes a fundamental discovery about human biology. Because the manager regulating the nerves factory isn't the nerve.
This is so wild.
It is. To find the regulator, the researchers had to look across the microscopic synaptic gap. They had to look at the muscle.
This blew my mind. We are talking about a transcellular communication axis. The muscle is actively managing the nerve that controls it. It feels totally backwards.
It really does.
Usually we think of the brain and nerves as the puppeteer, and the muscles are just the dumb wooden puppets waiting to be pulled. But here, the puppet is actively keeping the puppeteer alive.
It beautifully challenges how we view tissue isolation. We have this bias that organs and tissues are closed systems. But because the nerve is overproducing this protein, the researchers hypothesized that something from the outside normally keeps it in check.
Right.
And they discovered that muscles constantly secrete these structures called extracellular vesicles, or EVs.
Extracellular vesicle. Okay, let's make sure we can actually picture this. Think of an EV as a microscopic mail bubble.
I like that.
Yeah, so when a cell needs to send a secure package to another cell, it takes the information, wraps it in a tiny protective lipid bubble, kind of like a soap bubble, and pushes it out into the fluid between cells to be delivered.
That's a really great way to visualize it. And the researchers wanted to know exactly what kind of mail the muscle was sending to the nerve. So they analyzed these muscle-derived EVs.
Because lots of cells make bubbles, right?
Exactly. Every cell sends out vesicles, so they had to look at the specific protein markers on the outside of these bubbles to confirm they were the specific ones meant for this job.
Right. They look for tracking labels, basically. Things like CD63, CD81, CD9, and CHMP2A.
Exactly. Those markers confirm the origin. But the surface tags aren't the main event. It's the cargo inside the envelope that really matters.
If I get a package, I don't care about the cardboard box. I care about what's inside. What exactly is the muscle sending to the nerve terminal?
They cracked open these vesicles and found they were packed with something called the RNA-induced silencing complex, or RISC. And specifically, they carried a vital active component of this complex called Argonaute-2, or AGO2.
A go-to, let's translate that. In molecular biology, Argonaute proteins act like genetic scissors. Their entire job is to find specific strands of RNA, those blueprints we talked about, and cut them up so they can't be read by the factory.
Exactly. They're targeted destroyers, and the researchers actually managed to catch this mail delivery on camera, which is just a phenomenal technical feat.
I really want to paint a picture of this for the listener, because the setup alone is wild. They use these things called compartmentalized microfluidic chambers, right?
Yes. Think of it as a microscopic, perfectly controlled, dual-room apartment.
Okay, a tiny apartment.
Right. In one room, you grow human muscle cells. In the other room, you grow motor neurons. And between the two rooms is a solid wall with incredibly tiny, microscopic channels.
So they're separated.
Yeah, but the channels are so small that only the long axon of the nerve can squeeze through to reach the muscle in the other room.
Yeah.
It perfectly mimics the human body.
So using this tiny apartment, they essentially tagged the genetic scissors, the AGO2, with a glowing green fluorescent dye.
TFP, yes.
Right, and they watched the muscle blow a microscopic bubble filled with these glowing green scissors. The bubble floats across the tiny gap, touches the nerve ending, merges with the nerve's membrane, and physically dumps the glowing green scissors inside the nerve.
It's amazing.
They literally filmed the muscle mailing the tools to the nerve.
It is stunning to watch. And if we connect this to the bigger picture of disease, it highlights what we call a non-cell autonomous process. That means a cell's health depends entirely on a completely different type of cell. The motor nerve relies on the muscle to send it these regulatory scissors. Without the muscle's mail delivery, the nerve cannot control its own local protein factories.
But hold on. Scissors are useless if you don't know what to cut. We know the muscle sends the AGO2 scissors. But if the nerve factory has thousands of different blueprints floating around to build all sorts of proteins,
how do the scissors know to specifically target TDP-43?
Exactly. There has to be an instruction manual inside that envelope, too.
You've hit the exact next logical step the researchers took. To find the targeting manual, they sequenced all the tiny pieces of genetic code inside those muscle mail bubbles, and they found a highly enriched microRNA.
Okay, for those of us who aren't geneticists, a microRNA is a tiny, tiny snippet of code. It doesn't build the protein itself. Instead, it acts like a silencer. It binds to a larger blueprint and prevents the factory from reading it.
Spot on. And the specific silencer they found heavily packed inside these bubbles was called miR-126A5P. Right. And what was so crucial to notice was that this microRNA was heavily compartmentalized in the outgoing mail. It wasn't just randomly floating around the muscle cell being used for internal chores.
It was packed on purpose.
Yes. The muscle was specifically mass-producing it and stuffing it into envelopes meant for export. It was expressly meant for the nerve.
Okay, here comes my absolute favorite visual mnemonic of this whole deep dive. How does this specific microRNA, this miR-126A5P, actually stop the local nerve factory from printing TDP-43?
It's a great analogy.
I want you to think of the blueprint for TDP-43 as a zipper on a jacket. As the factory reads the blueprint to make the protein, it essentially zips the zipper up. The microRNA sent by the muscle is like a tiny, perfectly shaped piece of fabric that matches the teeth of that zipper at one highly specific spot.
Yes, in molecular terms, it binds with incredible specificity to the tail end of the blueprint, a section called the 3-foot untranslated region, or the 3-foot UTR.
So the piece of fabric jams the zipper. The nerve's factory tries to read the TDP-43 blueprint. The machinery hits that fabric jam at the 3-foot UTR. It gets stuck, and the whole assembly line shuts down.
Yep.
The molecular brake is successfully applied.
It's an incredibly elegant lock and key mechanism. But unfortunately, this beautiful system is where the pathology of ALS comes crashing back into focus.
Right, because things go wrong.
Exactly. The researchers looked at various ALS models to see what happens to this mail delivery system during the disease. They looked at mice engineered with human ALS mutations, like the SOD1, G93A, and G37R models. And they looked at the actual blood serum from human sporadic ALS patients.
And what did they find?
In all of these disease models, the levels of that specific microRNA inside those mail bubbles completely plummet.
The muscle stops sending the brake pedal.
Exactly. The mail bubble still arrives, the envelope is delivered, but when the nerve opens it, the specific instruction manual to stop building TDP-43 is missing, the brake is gone. And without that brake, the nerve's local 3D printer suddenly churns out massive toxic amounts of TDP-43, trapping all the other RNA, starving the mitochondria, and ultimately suffocating the neuromuscular junction.
Okay, let's synthesize where we are right now with our second quick recap, because we've just mapped an entirely new biological highway. Bullet 1, muscles send EVs to nerves. Bullet 2, EVs contain miR-126A 5P.
Yeah.
Bullet 3, this microRNA acts as a brake on local TDP-43 production. And 4, in ALS, the muscle just stops sending the brake.
That's the core of it.
It's a flawless theory. But I want to ask you, the listener, a question. In science, a beautiful theory isn't enough, right? Correlation isn't causation. Knowing this, how would you prove it in a lab? How do you show for absolute sure that stopping this exact mail delivery causes the disease?
That is the burden of proof. In science, to prove a complex biological system works the way you think it does, you have to break it intentionally. You have to sabotage the machinery in a healthy subject and see if it perfectly mimics the disease.
Here's where it gets really interesting. This is where the detective work gets brilliant. The researchers designed 2 specific sabotage missions. Let's look at the first one, which we can call the sledgehammer approach. They wanted to see what happens if they shut down the muscle shipping department entirely.
Right. For this, they used a genetic trick called a TET-on shRNA to knock down or turn off a specific gene in the muscle called RAB27A.
RAB27A, what does that do?
Without getting bogged down in the genetics, RAB27A is the loading dock door. If you turn it off, the muscle physically cannot release any exosomes. They also backed this up by treating the muscles with a chemical inhibitor called GW4869, which essentially acts as a chemical barricade blocking EV release.
So they put a permanent total hold on the muscle's outgoing mail. What happened to the healthy nerve?
It was incredibly dramatic. Remember how we said the toxic clumps trap RNA and starve the cell of everyday proteins? Well, when they stopped the muscle from sending mail, they measured overall protein synthesis in the nerve using something called OKP labeling. It's a specialized tag that lights up when general maintenance proteins are built.
And the lights went out.
That light almost went completely out. The nerve was starving. But then they used a different highly specific technique called PURO-PLA.
Wait, PURO-PLA?
It's a fluorescent tag that only lights up when a brand new TDP-43 protein is synthesized locally. And that signal went absolutely crazy. General production crashed, but local toxic TDP-43 production went into massive overdrive.
So it perfectly mimicked ALS.
Exactly. Consequently, the healthy nerve degenerated.
Which is an amazing finding, but if I'm being a skeptical scientist here, I'm pushed back. Shutting down the entire loading dock is a sledgehammer. Muscles probably send hundreds of different things to nerves.
Thousands, yeah.
Right. So that experiment proves the nerve desperately needs the mail, but it doesn't prove that our specific microRNA, the miR-126A 5P fabric jam, is the specific hero that prevents ALS. How did they isolate just that one instruction manual?
That is exactly the critique a peer reviewer would have, and the researchers anticipated it. They needed a scalpel, not a sledgehammer. So for the second sabotage mission, they didn't stop the mail bubbles at all. Instead, they used something called an antisense oligonucleotide.
Okay, antisense oligonucleotide is quite the mouthful. Let's think of it as a highly specific, custom-built molecular sponge. Let's call it miR-126I.
That is exactly what it does. They engineered this tiny sponge, this miR-126I, and introduced it directly into the fluid of the neuromuscular junction. This sponge was designed to specifically seek out, soak up, and neutralize only miR-126A5P.
So it leaves everything else alone.
It ignored every other piece of RNA, every other protein, every other vesicle. It just erased that one specific instruction manual as soon as the muscle mailed it.
So mail is delivered, but the sponge intercepts the brake pedal before it can reach the factory.
Yeah.
And what happened when they used the scalpel?
They saw the exact same pathology as the sledgehammer. By neutralizing just that single specific microRNA, the toxic TDP-43 factory spiked, general protein synthesis failed, the mitochondria starved, and the NMJ died.
Wow.
It was the smoking gun. It definitively proved that miR-126A5P is the crucial molecular brake keeping the nerve alive.
It's incredibly elegant, but a truly groundbreaking medical paper doesn't just figure out how to break a system, it tries to figure out how to fix it.
Yeah.
We found the missing brake pedal. Did they try putting it back?
They did, and this is where the hope in the data really shines. They asked the ultimate question. If lacking this specific microRNA causes the degeneration in ALS, can we therapeutically give it back and stop the disease?
Right.
They ran the rescue experiment in two ways. First, they used human stem cells. They took adult human cells, reprogrammed them back into stem cells iPSCs, and then engineered them with ALS mutations.
To make them sick.
Right. They grew these sick human nerves and muscles together. Then they artificially replenished the miR-126A5P.
And did it work on human cells?
It was profoundly neuroprotective. Replenishing that specific microRNA limited the TDP-43 accumulation and protected the human motor neurons from dying in the dish.
But a Petri dish isn't a body.
No, it's not. So they took it a step further and used viral vectors to deliver the microRNA back into the muscles of live SOD1G93A mice that were genetically engineered to have severe ALS.
This is the critical leap. Did giving the microRNA back to a living, breathing animal actually change the course of the disease?
It did. By restoring the brake pedal, it significantly delayed the motor decline in the mice. They held onto their muscle function, their ability to move, for notably longer. It didn't just look better under a microscope, it translated to preserved physical movement.
Okay, let's take a step back and really synthesize what this whole journey means for you, the listener, and for the future of medicine. We started this deep dive with the long-held assumption that ALS is strictly a central nervous system problem. The medical world has spent decades staring at the spinal cord, trying to fix the central power plant. But today, we've mapped an entirely new transcellular communication highway. We now know that your muscles aren't just dumb engines waiting for an electrical spark from your brain.
They're so much more than that.
They are active, vital guardians of your nervous system. They are constantly packaging up microscopic care packages, filling them with genetic breaks and mailing them across the synapse just to keep the local nerve factories from destroying themselves. If the muscle gets sick or the mail route breaks down, the nerve literally builds its own demise.
And if we connect this to the bigger picture of finding a cure, the therapeutic implications are massive. It is a genuine paradigm shift. It means we cannot hope to effectively treat ALS by solely delivering drugs to the brain or the spinal cord.
You've been looking in the wrong place.
We have an entirely new target. We have to look peripherally. The future of ALS treatment might involve targeting the muscles themselves. Perhaps we develop drugs that enhance their ability to secrete these vesicles, or we use gene therapy to artificially boost the levels of miR-126A5P right at the neuromuscular junction. It opens up an entirely new flank in the fight against motor neuron diseases.
It really changes everything about how we look at human tissue. We always tend to think of organs as these isolated kingdoms, The brain does the thinking, the heart does the pumping, the muscle does the pulling.
Right, in silos.
But they aren't isolated at all. They're talking to each other constantly, sharing source code, managing each other's local factories, and keeping each other alive in ways we are only just beginning to understand.
It's beautiful, really.
It is. Which leaves me with a final thought, a lingering question for you to mull over long after this deep dive ends. If we just discovered that our muscles are actively sending microscopic care packages of genetic material to prevent our nervous system from collapsing, it completely rewrites the relationship between tissue types. It makes you wonder, what other secrets, what other vital life-saving instructions are our muscles whispering to our nervous system right now, simply to keep us moving?